All right. Oh, I got the recording message. All right, everyone. So it is my pleasure to introduce today Dr. Alyssa Winter-May. Dr. Winter-May graduated from Colorado State University very recently in 2021 with her PhD in chemistry, and that was done under Dr. Travis Bailey. Her thesis project looked at functional nanostructured ionic liquid-based block copolymer systems for energy applications, which I know is very far out of the majority of our realms of expertise. And she's now the Soft Materials Postdoctoral Fellow in the Analytical Resources Core and Molecular Materials Analysis team at Colorado State University. And we have had the absolute pleasure of working with her and collaborating with her core resource facility. And we're happy to have her here today to present uses of the MALDI-TOF Biotyper and other resources at their core facility. So I'll have Alyssa go ahead and take it away.
All right, thank you so much, Rachel. I just want to thank you all for having me. This is really exciting. This is the fun part of my job, right? Getting to talk to people about science and the instrumentation that I get to work with every day. So it's really a privilege to be here. So yeah, we're going to talk about rapid identification of microbes using MALDI-TOF Biotyper. So a little bit about me, right? Like Rachel just said, I just got my PhD in chemistry in 2021. Working under Travis Bailey, who's actually a University of Florida alumni from the 90s in chemical engineering. We work with soft materials, block copolymers. So full disclosure talk about microbiology, I'm a synthetic polymer chemist. So bear with me. But I am a postdoc at the ARC MMA. We're a core facility operating under the Office of the Vice President for Research. So we're a centralized core facility with a ton of instrumentation that anyone on campus can come to use. I work largely in the mass spec lab. And so you can see this is me with my MALDI Microflex. So that's the instrument we're going to be talking about a lot today. The MALDI is one of my instruments. I focus on services for that instrument, training, maintenance on it. I've worked with some of the other LCMS mass spec in our lab as well, like our LC ESI TOFs and DART TOFs. But I also do work with other soft material analysis techniques. So mechanical testing, FTIR, potentially DLS, anything else related to polymers, soft materials. As you can see, we are at Colorado State. We're in Colorado. We love the mountains here. It's a great place to be. So if you're ever in Colorado, come visit CSU. All right, so what are we going to talk about today? First, I'm going to give you a quick overview of MALDI-TOF mass spectrometry. If it's a technique you're not familiar with, I want you, I always want people to understand how things work. Then we're going to talk about the particular method that I've been using to help Rachel analyze her samples, called MALDI Biotyper. We're going to talk about some applications of Biotyper technology, and especially Rachel is going to help me out and talk about the work that she's been doing. It's really cool. And then a little bit at the end, I'll give you a bit of an overview of the core facility I work at, the ARC MMA, some of our instruments and services. And then hopefully we'll have some time for some Q&A. All right, so overview of MALDI-TOF mass spectrometry. So if you're not familiar with mass spectrometry at all, mass spectrometry is a wide range of analytical techniques, but at their core, all mass spectrometers encompass a few key components, and that is some way to introduce your sample to the mass spectrometer, an ionization source, because all mass spectrometers require analytes to be ionized in order to be able to detect them. then some sort of ion sorting method, some sort of mass analyzer. Typically that's GC, TOF, quadrupole, all sorts of different techniques. Then there's always some sort of detector to detect relative intensity of ions, relative abundances, and then data system, well, that's our computer. So data output in mass spectrometry is usually relative abundance of ions in terms of their mass to charge. So anything that has a charge of 1, you're going to see the mass to charge as the molecular weight of that ion. If there is a charge of 2, for instance, like this little red molecule I have visualized here, the mass to charge is going to actually be half that of the molecular weight, of the full molecular weight of the molecular compound. So you'll see a lot of graphs in terms of mass to charge for the rest of this talk. So how does our particular MALDI-TOF work? Well, our way of introducing samples into the mass spectrometer is to put crystalline materials onto a stainless steel plate. That's called a target that gets placed into the target chamber. And then the MALDI ion source is really just a laser under vacuum. So this is our ion source. That's the MALDI part of it, the matrix assisted laser desorption ionization. And then the TOF part of it is our mass analyzer, a time of flight. At the end, there's a detector right at the top. So basically, this works by step 1. We mix our analyte, in this case, bacterial cultures, with some sort of matrix, which I'll get to what a matrix is a little in a minute, on a stainless steel target plate. We use a ultraviolet laser at 337 nanometers to desorb the samples and get them into the gas phase under vacuum. The vacuum is critical, so that way you don't have gas, various other gases like air interfering with the flight of your ions through the flight tube. Ionization occurs in a dense gas phase, so we'll talk about the ionization process in a little bit as well. Once our ions are in the gas phase, they're accelerated into the flight tube using some sort of high voltage. We measure the flight times of those ions as they hit the detector, and the ions will fly through the detector based on their molecular weight. So lighter ions fly faster, heavier ions fly slower. And then we correlate those flight times to mass to charge based on the calibration of known ions. In this case, our calibrant for bacterial testing is actually, they call it bacterial test standard, but it's E. coli. So what's a matrix? What's the matrix part of MALDI? This is an example of a matrix. It's alpha cyanophore hydroxycinnamonic acid, or we call it HCCA. So matrices are compounds that happen to be just the right molecular weight. So they're big enough that when you put the target plate in the vacuum of the MALDI, they're not going to just spontaneously evaporate into the time of flight analyzer without doing anything first. But they're small enough to volatilize when we hit them with the laser and initiate ionization. They tend to be acids, so they can be proton donors. Most MALDI methods are in positive mode, so we're initiating some sort of positive ionization, creating positive ions. So if they can donate protons to the compound, that's great. They tend to also be highly conjugated, and by conjugated, I mean they can have aromatic components like this phenyl ring here, or a series of double bonds where there's some delocalized electrons. Basically, electrons can hang out, so they can be potential electron acceptors, which is a very, very important component to these matrices, because the most important component to these molecules is that they can absorb UV radiation. So they're very strong UV absorbers. which is great because that means that they can absorb all of the energy from that UV laser, so your compound doesn't get completely obliterated when you shoot it with a laser. The reason why we need these matrices for the MALDI ionization process is because, so when we mix our compound of interest with the sample matrix and they co-crystallize on a plate, they dry, when they get shot with a UV laser, in this case a nitrogen laser, the matrix molecules are able to absorb all the UV energy instead of your analyte, so it doesn't get destroyed. Then those excited state matrix molecules transfer that energy to the analyte, which allows the analyte to go into the gas phase. And generally with MALDI, we're ionizing fairly large molecules that normally wouldn't be able to go into a volatilized gas phase. So the matrix is the key component to this, that transfer of energy is what allows our analyte to go into the gas phase without being destroyed. So we initiate desorption of the analyte, and then once our analyte is in the gas phase, collisions happen between the matrix and our analyte. Also, sometimes we had ionization agents like trifluoroacetic acid or formic acid that can help to transfer protons to the whatever it is you're trying to observe. And then once we have molecular ions in the gas phase, we pulse them with high voltage. It accelerates all the ions into the flight tube at the same time, and we get really good resolution that way. So I have a little video from Brooker that I'll play because I think it's really useful for visualizing the whole process.
The MALDI-TOF process is a two-phase procedure. Initially, the samples are fixed in a crystalline matrix. In the initial ionization step, they're bombarded by a laser. The sample molecules vaporize into the vacuum while being ionized at the same time. High voltage is then applied to accelerate the charged particles. The second step is the time-of-flight mass spectrometry phase. In the linear mode, particles will impinge upon the linear detector within a few nanoseconds after ionization. Higher mass molecules will arrive later than lighter ones. Flight time measurements make it possible to determine molecular masses directly. Each peak in the spectrum corresponds to the specific mass of the particle along the time axis, starting with the ionization moment. In the reflector mode, a particle...
We're going to cut it off there because there's a reflector mode, but it's not applicable to this talk because we only use linear mode for Biotyper, the Biotyper method. So what are some of the benefits of MALDI-TOF? Well, one, it's really low cost per sample. A lot of other mass spectrometry methods or bacterial identification methods require a lot of expensive reagents. MALDI is pretty limited on what reagents you need, and they're not particularly expensive, aside from the calibrants, but you don't need a lot of calibrants. So we can run MALDI-TOF samples for a few dollars per sample, really. It's pretty economical compared to a lot of other methods. We get really fast results. You can have spectra in seconds and identification in a matter of half a minute to a minute per sample. Minimal sample requirements, so you can get down to picomolar levels of concentration, super low. Higher is totally fine, but if you're really sample limited, we can work with that. Minimal sample prep. I'll show you sample prep in in a few minutes, but really just, can you get your sample onto the plate somehow? And then we can get it in the MALDI. Really great for samples with limited solubility. So other LCMS methods like ESI require your samples to be soluble in very particular solvents, and this has no solubility requirements other than can we get it onto the plate. We can measure intact macromolecules like proteins and also polymers. Really, anything within the range of that the MALDI can analyze. This particular MALDI can get up to about 200 kilodaltons. Another MALDI that we'll be getting soon can get up to about 500 kilodaltons. So we have a lot of flexibility on molecular weight. It's a soft ionization technique, so the MALDI process typically doesn't fragment samples, which is really nice. So we can observe whole proteins, whole polymer chains. It can produce multiply charged ions, but really not multiply charged, typically 2 or 3 plus at most. And then depending on what molecular weight range you're looking for, we can get mass accuracies as low as a few ppm. So it's really, really great technique for analyzing any sort of large molecules that are hard to analyze through other means. So there's a really high versatility of applications that we can use MALDI for. Today, we're going to focus just on microorganism identification. So let's get to the good stuff. Microbe identification using MALDI-TOF Biotyper. So MALDI-TOF, one of the reasons why it's becoming such a popular method in microbiology and microbiome research is that it's super quick. So if you're using a direct transfer method, basically colony straight to plate, it's possible to prep a plate in a full plate in under an hour, maybe if you're really fast, so half an hour or so. Rachel can speak to that more than me. But straight from colony on a plate, wipe it on a spot, drop some liquid matrix on it, throw it in the system. We can analyze the full plate in about an hour, and results. It acquires spectra and analyzes results as it goes. So by the end of that hour, it's already identified everything. Pretty incredible. So this is just a good friend of mine works for the microbiology department at UC Health, which is a local hospital in town. And so she made these great videos for me of their MALDI-TOF sample prep process, because she runs MALDI-TOF every day, all day at work for doing clinical diagnostics for the health system, hospital health system. So as you can see, she literally picks up a colony with a toothpick and fills it into a circle on the plate. Fills it into a circle on the plate like a Scantron with a toothpick. It's as simple as that. Next, she takes the matrix solution, drops it on, and lets the spot dry. I know Rachel will often do a formic acid digestion on her spots, because that can help lyse the cells and help them ionize easier often. So that's also an option, but it's also just drop some formic acid on the plate. It's really simple. So what do the results look like? Well, it's like I mentioned earlier, it's relative abundance, intensity compared to mass to charge. So this is a mass spectrum for E. coli. And it's a really good identification method because it relies almost entirely on very highly abundant proteins in cells, typically ribosomal proteins, which tend to be hydrophobic and dissolve really well into the organic solvents that the matrix is dissolved into. So when you place the matrix onto the plate, the organic solvent in that mixture actually will dissolve and co-crystallize the ribosomal proteins with the matrix. We only look for the range of 2,000 to 20,000 daltons. That's really all it needs, but that's a range that ionizes really well in this instrument, so it's more than sufficient. Okay, so how does the instrument go from a raw spectrum to hey, this is E. coli or other bacteria or other microbes? Well, what it does is in the background, it matches the sample spectrum to a whole library of reference spectrums, and we'll call those main spectrums or MSPs. So for instance, this top spectrum would be our sample spectrum. The bottom set of peaks would be the MSP and the library, the set of reference peaks. And as it's in the background, it's matching peaks. So it says, okay, there's 6 matches here. So it's going to say, it's going to calculate 3 values. One is going to be how many of those matches are compared to the reference spectrum. So in this case, there's 6 matches to the 10 peaks of the reference spectrum. And that's going to get a value of 0.6. Then it says, okay, how many matches are there on my sample spectrum? Well, in this case, it's 6 out of 20 unknown spectrum peaks. And so that's going to give us a 0.3. And then it gives a correlation of how well do the intensities of those matching peaks match. That math is unknown to me, but it gives a value of that intensity correlation. Then it takes those 3 values, multiplies them, adjusts that to a value of 1,000, and then takes the logarithm of that, which since so maximum, if the score was 1,000, we get a maximum score of 3. If the score is 100, you get a maximum score of 2, since it's a logarithmic scale. So anywhere between 2 and 3, we have a pretty high confidence identification, and you get a score. So for instance, this is an example output for a particular sample with pretty confident identification of this particular microbe in the 2 range, and then gives you some other microbes that are a particularly good match. So for every sample, it'll give you a list if you want. So what kind of microbes can we identify using the Biotyper? Well, there's a library of about 10,000 different microbes. The biggest library of that is the bacteria and yeast library, but there's also a library for filamentous fungi and mycobacteria.
Alyssa, can I ask a question?
Sure, absolutely.
How often do they update their library? So curated to new species?
Bruker puts out an update every year.
Okay.
Yeah.
How much have you seen the growth? You know, so you're at 8,000 right now or something?
It's somewhere, yeah, somewhere in the high 8,000s. Yeah. I will say I don't know. I only started working with this instrument a year ago. So, but I do know they're constantly adding to it every year. So
Okay.
I think since it's a fairly new thing. I think, only within the past 10 years, I can only see it kind of getting
Right.
better and better every year.
Sure. Thank you.
Yeah. To add to that point, though, a lot of people are working on building their own custom libraries. One of the bigger complaints that I've seen looking through research in the microbiome biotyper field is that some of the stock manufacturers provided libraries are more clinical laboratory focused. And so there are a lot of researchers out there building their own libraries to disseminate to other researchers. So we can build a custom library. We have that capability. So this is what you're looking at here is actually my attempt at building my own custom library from some random spectra that I acquired from Rachel's plates. So I took 3 spectra, turned them into MSPs. So that's what you see here, test MSP 1, 2, and 3. Test MSP 3 is a BTS sample, the E. coli bacterial test standard. And so what you see here is actually me running a identification against my own custom library of these 3 samples. And this is actually BTS matched to itself. So that's why the peaks match really well. It gave me a score of 2.85. For some reason, it didn't give me 3, even though it's the same sample. But what you'll notice is that it gave me a really bad score for the other two, which I think is good. We also have the option of when we build a custom library, giving it an organism name, strain name, extraction conditions, matrix, growing conditions, all sorts of that stuff. So you can really put in a lot of information into the custom libraries. There's a lot of flexibility here. So I think it's really great that the manufacturers built that, knowing that their libraries are inherently limited by what they're able to compile. You have that flexibility for when you need it. Okay, so what are some applications for the MALDI-TOF Biotyper? Well, my friend Jackie, who gave me all of those great videos about the MALDI-TOF sample prep that she did, gave me also some interesting stories about how it's really revolutionized clinical diagnostics and microbiology. And this is one example she gave me of a study where they were developing methods for pelletized blood cultures, so really quick, about an hour or two, methods to take from sample acquisition to culture. It was about 2 hours to acquire that sample, and then they were able to identify the bacteria in those cultures essentially instantly, within about 15 minutes after they did the pelletized culture. Whereas in prior pre-MALDI-TOF methods, they would take over a day to gram stain and incubate and culture and do a lot of other microbiological workup. Another study from Johns Hopkins studied the cost savings and time savings for their clinical microbiology lab over the course of a year, and they found on average they got results to patients a day and a half faster, which is really huge for for patients that require immediate treatment, right? So it also reduced the reagent and labor costs by about 60%, which is really incredible. Another application, this wouldn't be a proper Colorado talk if we didn't mention marijuana. Microbial contamination in cannabis is actually a big problem. Greenhouses are warm and humid, really great environments for growing bacteria and fungi. So most states that have legal marijuana, that have legalized marijuana, require QC testing for microbes. And right now, I don't think the industry is particularly aware or involved in using MALDI-TOF. From what I understand, a lot of their methods are culture, pretty standard traditional microbiological methods, ELISA, PCR. So they're cheaper up front, but reagents can be expensive. There's also other potentials, false negatives, positives, other disadvantages, a lot more labor costs than that. So, we really think that MALDI-TOF Biotyper could be huge potential for cost savings for that industry. And so that's one thing we've got a lot of thoughts for marketing our laboratory services to that industry, particularly in our state. Since even though the upfront instrumentation costs are high, it's really cheap to run, faster results, more reliable. So since we already have the instrumentation, we think that can be a good market for the industry in this state. So there's actually a lot of MALDI-TOF Biotyper being used in microbiome research at the moment. People doing large-scale worldwide culturomics studies and metabolite analyses. It was really cool to see all of this in the literature recently. But now I'm going to hand it over to Rachel, who's been so awesome in explaining all of her work to me. But I can't do it justice, so I'd like her to talk to you about why we're working together.
Yeah, so thank you so much, Alyssa. I really feel like I understand a lot more about what you do on your side. So thank you so much. So today I'm going to talk about the project that we are using Alyssa's help on to build kind of a custom consortium of bacterial isolates. And this would be pretty much impossible without the use of the MALDI Biotyper and Alyssa's expertise. So I'd like to speak to that. So my project focuses on examining microbial drivers of response in patients who have cancer to immune checkpoint blockade. And specifically, we are looking at a subset of patients who were treated at Moffitt Cancer Center with immune checkpoint blockade that had non-small cell lung cancer. And so what we did with these patients is we collected baseline fecal sample and post-treatment fecal sample, and we found through 16S sequencing that these patients have a different microbial community structure at baseline. And so Alyssa, you can move forward. And so we had also collected some of these baseline patients from responders and non-responders in LDTM preservative media. And so what this enabled us to do was to humanize a gnotobiotic mouse model of Lewis lung carcinoma treated with PD-1. And we were able to stratify these mice into responder, non-responder based on the fecal implant they got. So half the mice got a fecal transplant from responder patients, half got one from non-responder patients pooled together. And these mice recapitulated the response from the original donor. And we performed 16S sequencing on the output from these mice. And Alyssa, you can move forward. What we found was very, very interesting. So we were able to find specific genera and taxa that were both enriched in responders or non-responders. But what was curious to us was that each of these points on this log fold change plot showing taxa enriched on the responder side in left and enriched in the non-responders in right shows the presence of different microbial isolates or amplicon sequence variants, as annotated by our sequencing data, were differentially abundant in our responders versus non-responders. So if you look at the genera Bacteroides, you can see that there are ASCs enriched in responders, but also ASCs enriched in non-responders from the same genera. And so this told us that we are not just looking at broad changes in microbial community structure, but rather there may be gene content of specific isolates that are driving this phenotype. And so what this drove us to do was to try to find these specific isolates and specific strains of these genera that were driving response. And so Alyssa, you can move forward. So the pipeline that we developed in collaboration with Galt, who did speak in this microbiome working group about a year ago, and Alyssa, was to do a high throughput identification of human fecal isolates. So what we were able to do is generate thousands of individual unique isolates from input responder feces using the Galt Prospector platform. But the issue we came into was how do we identify all of these isolates? 16S sequencing is effective, but extremely expensive, coming in at about $5,000 for a set of maybe 200 isolates, maybe a little bit more. It would also take a very long period of time. And so we reached out to Alyssa because we actually don't have this capability here in our core labs at UF to see if we could possibly use their Biotyper facilities. And so what we do now is we send all of these isolates in 96 well target plates to Alyssa. We ship them overnight, and then she runs them for us. And what this has enabled us to do is generate a huge library of identified isolates that come directly from our fecal samples from our responder mice, allowing us to look for taxa that may be of interest to us based on our original 16S data. And so Alyssa, you can move forward. So one change from, I guess, the traditional Biotyper workflow is that all of our isolates are anaerobic, so strictly anaerobic, and they are all grown in liquid culture. So this was a little bit of a challenge to us, but it turned out not to be at all. All we do is we grow our 96 well plates of isolates in a deep well plate. We just spin them down wash the pellet with water, and we're able to load that bacterial pellet on the target plate just as if it was a single colony. What we do know is that each of these are unique isolates without contamination, and so that enables us to do it this way. Otherwise, you would need to, of course, streak a plate to get an individual isolate. So we're very glad for that. And then moving forward, Alyssa, how our workflow is now is we prepare all of our isolates inside our anaerobic chamber, and we actually do all of our streaking of our plates and the addition of the matrix and such in our chamber. And then at that point, we take it out and ship it to Colorado. So we do a 5-day culture of our strict anaerobic isolates, pellet and wash, as you see in the chart on the right. We streak these in duplicate, actually. And as Alyssa mentioned, we usually apply formic acid to one of the duplicates and then matrix on top of that, and then just matrix on the other. And the formic acid just gives a little bit of a better extraction on some of those really difficult to identify isolates. So it does give us the benefit of having a little bit deeper extraction on those isolates. And so we just do it in duplicate, and then we send that to Alyssa overnight, priority on FedEx on dry ice. And as you can see, the little MALDI in the mountains, she runs them next day in Colorado. And so Alyssa, you can move forward. So just in this short period of time that we've been working with Alyssa, we've been able to identify hundreds and hundreds of isolates just using her system. So as you can see, we do get a lot of Bacteroides ovatus, and I'm sure we would all be very depressed to send off a 16S sequencing run and only get Bacteroides back. So the cost per sample makes it attainable to actually identify everything as opposed to hunting down specific libraries. And so we have here about 30 unique species that we've identified. And if you move to the next slide, Alyssa, we have total, as of now, about 700 isolates that we've identified with their group. And as you can see in the pie plot to the left, we have an excellent profile of different genera, even different families that we've gotten from the MALDI, which we've also been able to pull into a consortium that we've identified with 16S, that everything kind of checks back to the original MALDI identification. And now all of these isolates are available to us in the lab to use as human fecal isolates from a clinically relevant sample population in our mouse studies and in all of our other research. And so this represents a hugely valuable tool for us, and really has kind of pushed my project into the next stage of its life. And we couldn't have done this without the MALDI core. And so that kind of concludes my work with her, but I hope it really speaks to the abilities of the MALDI Biotyper and the usefulness. I think maybe we've only spent about $1,000 or $2,000 total to get all of these identified, which is just a fraction of the cost that even just one 16S run would cost. So we're really happy with what's been done.
All right, thank you so much, Rachel. Yeah, I think it's been about $2,500 since July, was it? July? Yeah. Okay, so because I work for core facilities, and I have to give the core facility shameless plug about our instruments and services. So first off, since Biotyper is what you're all here to hear about, our current rates for the Biotyper, if you send us a pre-prepped plate the way Rachel does, which is super fantastic, we really appreciate that. It's only about an hour of instrument time, about 30 minutes of staff time to run a single plate. And Rachel does hers in duplicate, so I think it's somewhere around 44 samples per plate. But if you didn't want to do that, you could identify theoretically up to 95 isolates for about $100. So, I mean, that's just up to you and your workflow. I do think doing duplicate is good, the way she does it with the formic acid, not formic acid. I think that gives her probably more reliable results, but you know, that's up to the customer really. Oh, we do charge a once monthly administrative fee of $78. So, you know, if you were to start sending us plates, I don't want that to be a surprise. We can do plate prepping services. We have a sister facility, our bio facility that has laminar flow hoods and that sort of expertise. So that could be an extra charge. I'm not a microbiologist, so I will not be prepping any Biotyper plates, but we can do that for you. Sending from Florida, that would be a little tricky, I think. So probably be better to just prep them yourself. The plates, I think, are only a few $100s. I don't know, Rachel, how much are they? Yeah, the stainless steel plates from Brooker. So you can just buy those from Brooker yourself and ship them to us, and we just ship them right back to you when we're done. So we do prefer the plates prepped, shipped to us. That's a lot. That we can get your results much faster that way. So I work in the mass spectrometry lab. We have a pretty large variety of instruments. So I just want to give you a little preview in case you were interested in any other sort of full service analyses through us. We do full service projects of wide variety for all sorts of different customers all the time. So we have 3 GCMSs, a GC single quadrupole, a thermal desorption GC quadrupole, which is great for atmospheric samples, concentrating atmospheric samples. And we have a GC triple quad for quantitative analysis. We also have a UPLC triple quad, which is great for targeted quantitative analysis of all sorts of different compounds. We have two TOF instruments, one Bruker Maxis UPLC Q-TOF, which is super high resolution. It's a really excellent instrument. It's a quadrupole time of flight, so it can do fragmentation and structural elucidation that way. We also have a UPLC single TOF, which is great for accurate mass, but the cool thing about it has a lot of flexibility in terms of ion sources. So we have 3 different possible sources: ESI, APCI, and DART. DART is really cool for, you can literally stick your sample straight in it. I've actually stuck a habanero in it one time. You can basically analyze anything in the DART. And then, of course, last but not least, our MALDI collection that it's about to be. We have the Biotyper right now. That's the instrument I talked about today, but we just got an NSF MRI grant funded to purchase a Bruker Ultraflex Extreme MALDI-TOF, which we're really excited about to serve a wide variety of people on campus and off campus, particularly our synthetic polymer chemists that struggle to ionize synthetic polymers on the microflex. So the analytical resources core, I mentioned it before. It's a collection of 3 core facilities under the OVPR's office, the Office of Vice President for Research. I work for the MMA. We have a lot of our materials and molecular analysis type of instruments geared towards chemists and other people, mass spec, NMR, spectroscopy, X-ray diffraction. We can do crystal structures, all sorts of things. The imaging and surface sciences focuses on microscopy. They're really great. They're working on getting cryo-TEM set up here, which will be really exciting. And then our bio facility, bioanalysis and omics, there are bio experts, and I know all sorts about biology that I don't know anything about. So they're our go-to for metabolomics, proteomics, ionomics, all that sort of stuff, any sort of bioinformatics work. And with that, I want to thank everyone so much for attending this talk. It's really exciting. This was my first invited talk as a PhD. Of course, Rachel Newsom, I wouldn't even be here without Dr. Christian Jovin's lab and Rachel Newsom's work. So this is really exciting for me. Thank you to Galt and all of your collaborators at UF. And of course, Jackie, my friend who works at UC Health, for giving me those videos and telling me all about microbiology things opening me up to the to the world of microbiology. And my art colleagues, of course, my bosses, Caroline Deneff and Claudia Boot, they're both super fantastic. Kitty Brown managed the MALDI before me. She's the one who trained me how to use it. She's my colleague at ArcBio, who actually the MALDI microflex was actually at ArcBio first, and then the instrument came to us about a year ago. And then Annalise and Clayton have our undergraduate student hourlies who have helped me run many plates for Rachel, particularly during the month that I was going crazy writing my dissertation last fall. So, and that's it. I would love to answer any questions you may have. I'll leave up my contact information while I'm answering questions, if anyone has anything, and please check out our website. And if we end up doing any service work for you, please acknowledge us by our research resource ID. It's an ID for core facilities all around the country. Yeah, thank you so much.
Thank you,
Thank you,
Alyssa.
Alyssa.